The other one is hemoglobin concentration, where you're actually putting your own blood. So this week you're going to be dealing with blood, so please, you don't need to wear gloves, and it would be very hard to prick your fingers and take blood with a glove on. But as long as it's your own blood, it's cool. You can touch it, you do whatever you want, as long as it's your body that it's contacting, or one of these plastic dishes. Just please don't touch other people's blood. But what you can do is just put your own blood on a very, very white piece of paper and basically compare its color to get a rough estimate of your hemoglobin concentration. And yeah, so that's pretty much it for our blood tests. So blood typing, that is probably the hardest thing we're going to deal with today. Not a whole lot of terms with blood typing. Well, let's just take a look at this chart real quick. Since you guys have already talked about blood, you guys have thought about this already. However, this is a very difficult topic for a number of different reasons. The biggest one is that you know how you only really read like the beginning of words when you're reading stuff quickly? Well, it's antigens and antibodies. So it's very easy to exchange those two things, and they're opposite concepts. And so you have to be very strict with yourself and how you talk in your own head. Because you know, you rationalize things while you're studying in your own head, and you need to make sure every time you say antigen, you mean antigen. Every time you say it to yourself. Because you can study all night flip-flopping these terms, and you'll learn the whole thing backwards. And so let's first start by what you think of when you hear antigen and what you think of when you hear antibodies. I love to use symbols, and so my symbol is very similar to what you see up here. When I think antigen, I always think of a circle with something sticking off of it. Because an antigen is just an external feature of a cell. So it's just like a finger sticking out of it. Like this could be one of my antigens, would just be my index finger. It's just the shape of the outside of the cell. Now the function of the antigens are much like an ID badge for your immune system. If you have an ID badge that says, hey, I'm supposed to be in Greg's body, then your immune system basically ignores those cells. But if you have the wrong antigen on the outside of the cell, then the immune system will attack. That's how it's built. That's how it knows, even though me and a cow are made of identical types of tissue, if any of their antigens get into my blood, my immune system will recognize the difference between me and the cow's blood. And so that's all based on antigen. Now antibodies, those are your Y-shaped things. So these are proteins in your blood, and they attack a specific antigen. So for every antigen, there is in the world, not necessarily in your body, but in the world, you could build a antibody for that antigen. Now, in our bodies, we are actually trying to avoid this situation right here. We do not want to have that antigen near that antibody. Because if these two are near each other, they go through a process called agglutination, which is what you see in the top right. It's basically the antibodies. So in this case, it's the red antigen with the red antibody, and those antibodies are binding up all of the A-type blood in this example, or the ones with the red circles on them. And so that's because, in this example, the invader is the red A-type blood. So we're actually trying to avoid that situation when it comes to blood transfusions. We don't want that to happen. So normally, you don't want these to mix. But if they do mix, that's going to happen, and that's usually a good thing for your immune system in terms of finding and keeping those cells close together. But for a blood transfusion, you clearly don't want that to happen. So we're going to talk about how you can avoid that in a moment. So antigen, describing the cell. Antibody, describing a protein that's in the plasma. So that's step number one that makes this kind of confusing. Now the next part that's kind of confusing is how we talk about blood types. So as you can see, we've got O, A, B, and AB. You guys have talked about the Rh group. That's another blood antigen. It's another thing on the outside of red blood cells. It's usually discussed in a different manner because with these blood types, they're very common. Like everybody has these. There's not people that are left out. The closest thing to being left out is O, but in reality they still have antigens. This is just like a way to think about it. There is an O antigen. It's not actually a naked cell, but it is an invisible cell to the immune system, so it's effectively naked, like it's illustrated here.
It's a ninja.
It's a ninja, yes. It is behind the scenes. Now, the antibodies, though, you guys talk about how you develop antibodies? So it's mostly based on exposure. You have some innate antibodies. You can get it during fetal development. You definitely get it during breastfeeding. But for the most part, you got to be exposed to stuff. the AB blood groups are so common that everybody, either through development or through their actual living living human lives, will be exposed to these so early on that we always assume that if you have this type blood, type A, then you will not have type A antibodies because they would attack yourself. You don't want that. But we assume that you've been exposed to this B antigen at some point in your life, and therefore you've developed B antibodies. So the development of antibodies is based on your prior exposure. For almost every type of antibody we have to be exposed to it. So that's the basis of vaccines, right? You give a little bit of exposure, your body recognizes this invader and it learns and remembers and constructs antibodies so that the next time that thing comes, there's a vicious response before it actually hurts you. So this is what this second column is trying to illustrate. What antibodies are present in that person's plasma? And hopefully it makes sense that you would only want B. You do not want A because you would attack yourself. And then here's the opposite scenario right here. And then here's our AB scenario right there, where you can't have A antibodies because you have A red circles on your cell. Can't have B antibodies because you have the blue triangles on your cell. So if you had the B antibodies, you would attack yourself. So someone who's AB type blood like me would have the fewest antibodies when we assume exposure. So I want to point that out because there may be situations where people haven't been exposed before, but you're always going to be conservative and you're going to assume exposure. So for any question I ever ask, if I give you a blood type, A, you just assume that that same person has been exposed and has the anti-B antibodies. RH works in the same way. It's a third class. It should be just C, but it's not. It was discovered in rhesus monkeys in the 40s, and it got into our genes way later, so we don't have quite as vicious of a response to it. And the exposure is not nearly as assumable, but for this class, if they don't have RH antibodies on the cell, we will assume that they do have RH antibodies. But that's the only one I'm sort of lying about. But it makes the logic a lot easier to follow. So what I'm going to show you is how to, this is a lot to take in and it's something that takes a little bit of practice. And so I'm just going to show you how I use logic and reason to get to an answer here rather than like trying to memorize, oh, an A negative type blood can accept, these 4 different types of blood. And I'm really visual, and I know a lot of you guys are too, so I think it's really helpful to stop thinking about this in your head. You need to have something you can actually visualize. So what I do is something called the blood box, and it's just a box with a circle in it. So it's very simple. You can use it on your quizzes, on your exams. It makes it very easy to visualize what someone's blood situation is. So let's start with 2 extremes. So all it is this. The circle's the red blood cell. The box is just to enclose it in something. The white area out here is the plasma. So it's really what we were looking at on the other picture, but in a little bit more concise way. So I'm going to do 2 examples on opposite ends of the spectrum. So first one's going to be A, B positive, and this one's going to be O negative. So that's me here on the left. And remember, antigen is the shape on the outside of the cell. And the other thing to add into that part of your memory is that when someone tells you their blood type, it's describing what their red blood cells look like. It is not directly describing their plasma at all. So when I'm AB positive, all I'm telling you is I have the A antigen, the B antigen, and then I'll draw a little RH to represent the plus or the minus. So positive means the RH is present. If there was a negative there, it would not have an RH on the cell. So that's what AB positive means. It means I've got a cell with all three of those antigens sticking out of it. Now, the blood box also, first you draw it yourself, and then whichever one of these three is not on the cell, we assume is out in the plasma as an antibody. However, all three of them are on the cell, so there's no more antibodies to put in there. We can try it just to sort of prove it to ourselves. So if we do this, an A antibody, that's a bad situation. That's not how I am, because these two would match up with each other in agglutinate. And then you can try it with a B and try it with an RH, and hopefully you can visualize why that can't happen. Because you can't have two A's, two B's, or two RH's within one box. So I'm done here. I have a very friendly plasma. This is why this is referred to as the universal receiver. AB positive, in terms of these three blood groups, it does not matter what other types of foreign blood you put in there. It could be an A type, no problem. I can take anything with B, no problem. You can even throw an RH on there. It doesn't matter because that's all self to me, because all three of those are on my body. Now, opposite end of the spectrum, you can probably see what's going to happen over here. We're done drawing our red blood cell, because O represents the absence of either A or B, and the negative represents the absence of the RH. So that is, in that sense, kind of a naked cell. So, well, we're not done because we still have to put an A, B, and an RH somewhere. Try to be consistent with my Y's. So that means we assume that this person's been exposed to both A and B and RH. So at some time previously, they were exposed to these in their blood. They developed the antibodies for A, B, and RH. So this is not the universal receiver. This is the hardest person to give blood to. So the only type of blood cell that can be given to this individual is another O negative. Because if you're O positive, you're almost naked, but you've got an RH, and you're going to have an agglutination reaction between those two. So the situation for O negatives is definitely a lot harder than the situation for AB positives. They're opposite ends of the spectrum. Now the good part is that this O negative type blood can be given to anyone. That's why it's such a coveted type of blood when it comes to donating blood. And so because it has no recognizable antigens on the outside of it, you can give it to anyone. My blood, virtually worthless. Just donate plasma, they say. Because my plasma is actually empty, which is a good thing. It's kind of a blank slate. So those are our two opposite ends of the spectrum. Let's just do one in the middle just to kind of practice this same thing. So let's do A positive. So again, just pause and think, what does it mean to be A positive? That's describing the outside of the cell. A and RH antigens. And that leaves only one left to be illustrated in your box, and that's B. So now from looking at this middle one, you can kind of see what you want to avoid. If this is you, you don't have to memorize a list of blood types that an A positive person can receive. All you have to do is avoid B, because that's the only thing that this person's going to react with. So any blood type with a B in it, you can't donate, because you're going to have agglutination. But if it doesn't have a B in it, you're good. Does that make sense to everyone? And so that's just really, I found a much nicer way than like trying to memorize it. You can just sketch this on the back of an envelope or something like that when you're trying to reason through it in your head. And it's really just three different things that are going to be distributed in a lot of different ways. So I'm going to ask you to tell me, if I give you a blood type, if I give you AB negative, what type of blood can that person receive? That's one way that I can ask the question. So let's just do that right now. Just get some more practice. So A, B, negative. That means an A and a B on the outside of the cell. And then we've got the RH out there. So just from looking at that, we know we have to avoid anything with a positive on it, right? Nothing with a positive. So you're fine to have A negative, B negative. Obviously AB negative, you know that from the beginning because that was the original blood type. You can have O negative, and we're good. So that's the process you're going to go through for that. So you can work through it in that direction. I give you a blood type, and I ask you what types of blood can this person receive. The other thing I could ask you, or the other way I could present it, is we are actually going to do blood typing. And so you're not going to all be able to see it, so I'll just draw it up here. But you're given these little plastic plates with little wells inside of them. And you're going to put a drop of your blood in each one of those little circles. Then you're going to mix 3 different antibodies into these wells. So here's what they look like. So you're going to put as much blood as possible, so you're going to prick your finger, and you don't have to do this. If you don't want to stab yourself in the finger today, it's cool. But at least watch somebody do it and make sure you can interpret these, because the other way I could ask you is I could give you the results of the blood typing and then ask you this question. So you have to do 2 different things. You have to interpret the blood type, and then you have to go through the steps that we just went through. So let me give you a couple examples of just what this might look like. So it's labeled as A right here, B down here, and RH up here. So your blood, you're going to have hopefully a good sized drop of blood inside of each one of these. The more blood, the easier it is, but you really can do this with a small amount. It's just a little harder. You're going to have 3 identical drops of blood, and then we have 3 different antibodies that are in the freezer right now because they have to be small. We have 3 different ones. 2 of them are different colors. One of them is anti-A, the other one's antibody B, and the other one's antibody RH. And you're just going to take a drop from the A and put it into this blood. So this is the antibody for type A blood. So if your actual blood type is A anything, you will actually have agglutination. And so if those of you who know your blood type now, if you have an A in it, then you should expect to see agglutination in here. And what that looks like is your blood clumps together in little clumps, and you can't mix it back together, so you have little toothpicks to try and mix it. If it doesn't mix back together, then you've seen agglutination. And then you're going to do the same thing with B, putting it in here, and the same thing with RH, putting a drop in there. And so, for me, because I'm AB positive, I would expect to see agglutination in all 3 of these wells. So when you see agglutination, that means that is your blood type, or at least one component of it. And then if you're A negative, it would just be agglutination in A, but these would still look normal even when you add the antibodies into it. Does that make sense to everyone? Mm-hmm. So you're creating the situation you never want to happen in real people. You're creating that over here to figure out someone's blood type. All right, so any questions about this? All right. So I think that's pretty much all I want to talk about together as a group. I want to mention one more thing about the blood slides, about the microscope work you guys are going to be doing today. Oil immersion is an option. So we got oil immersion for those of you, most of you have done this before at least once in Biology 112 or in microbiology many times. So oil immersion is only for the maximum, the largest lens that we probably, hopefully never use because it doesn't actually help you unless you use oil. This lab is always written as if people are going to use immersion oil all the time. I just recommend that you actually don't have to use it, but you're going to want to get closer, and you're going to need the oil to do that. But just try to do it at the end of your studying, because once you put the oil on, you can't really go back. Because you put the oil directly on the slide, and once you've done that and you want to zoom out, you can't do it. And remember that as you get closer, it takes longer to search around the entire slide. So I would recommend putting off the oil immersion until later, or if you're at a group of, if you're working in pairs, just use the oil on one of the slides, because I really want you guys have to clean this stuff up. It's messy. It's oil. You need to use the detergent that's inside of our little yellow bins and wipe it off at least twice, if not 3 times, to make sure all the oil's off of the big lens. And also wipe off the slide. Don't forget about that. So I personally don't think you have to use oil immersion, but you're going to want to be closer. If you feel like you're close enough with the second to strongest lens, then screw it. You don't have to use oil immersion. That's fine. But just realize that once you put it on, you can't really go back unless you want to clean it up and then go back. Please put your microscopes away so in a really organized fashion. Make sure that the stage is down all the way, the lowest magnification is up, and that you've cleaned it up awesomely. This is a relatively light week in terms of material. It overlaps a lot with lecture. So lecture's caught up with us, or we've caught up with lecture, either way. And the next two classes are going to be on blood vessels and the heart. And I know you guys started the heart, right? So if you would like, I think if you're trying to get ahead of the game for this second half of the term, I'd recommend maybe starting the heart today, like just get a basic outline of the different chambers. Take a look at our giant heart model right back there, and there's more models in the back center of the room. Try to get ahead of the game, because this is our lightest week for the rest of the term. And so it's a good, if you're trying to get ahead of schedule, this is probably the week to do it, because yeah, this stuff today probably won't take you too much time. But I encourage you to use that time to get ahead of schedule. All right. So let me get